about something. You know those bacteria lists you all handed in?
Yes.
How come no one knows that Clostridium perfringens causes gangrene? Everyone put food poisoning. There is such a thing as gangrenous food poisoning. You can get food poisoning. I can't even talk there. You can get food poisoning. I said food poisoning. Whatever. Food poisoning from Clostridium botulinum. Also, Clostridium perfringens. There is a gangrenous form of food poisoning.
I'm looking at it right now. I did put gangrene, so.
Right.
It's not that no
Most
one-
people only put food poisoning. You need to know your Clostridium for this test. Clostridium perfringens is gangrene. Clostridium botulinum is botulism. Clostridium tetani, tetanus. Clostridium difficile, C. diff. Clostridium perfringens does cause a type of food poisoning. But even more, at least as equal to that, is that it causes gangrene. So you need to know Clostridium perfringens causes gangrene. Very few people put gangrene on their list. So what were you using for a reference that said food poisoning? Does anybody remember? That's your homework assignment for tonight. Go home, look at your list, see if you wrote Clostridium perfringens causes food poisoning, or gangrene, or both, and then tell me where you got that reference from. Was it from the text? Huh? CDC said only food poisoning, didn't say gangrene?
I have my notes that you talked about it, about really laying out too long, or being like, crooked.
That's botulinum.
Which one do you have?
Talking about botulinum food poisoning.
Which one do you
Gangrene does cause food poisoning, but also gangrene. Yeah.
I think the reason
Yeah,
I
but where's,
specifically put food poisoning is
where's your
because it's
gangrene
the
at?
most
You have
common
anywhere?
cause of food
No,
poisoning in the
I do.
US, so I felt that that was important.
Gangrenous food, yeah. CDC said that?
Yeah.
Yeah, that's weird, I wouldn't think so. Here's another one I got a lot of. Treponema, what does treponema cause? Treponema pallidum, what's that cause?
Syphilis.
Syphilis. A lot of people put visible, only visible using dark field. A lot of people said treponema was only visible using dark field. So what reference did you use for that? It may well be true, but I kind of doubt it. It's probably easier to see using dark field. If you stain a spirochete, it doesn't really stain well, first of all. You know how most cells have a natural negative charge and attract the direct stain? Treponema doesn't do that. So they have to use a special stain with silver in it to penetrate treponema to make it stain. And you can see it in a special stain preparation. If you look at it in a wet mount, you'll see it in its spiral shape, and that would be dark field. But I have seen stains of it. In fact, I think I showed you slides of it. And it's stained black and it's in yellow tissue. So I don't know about that reference, visible only in dark field, because there are staining techniques you can stain it with, in which you would use bright field. And here's something else about your list. A lot of you put descriptions of the disease, the name of the disease, beautiful, perfect, but you forgot special characteristics we talked about in class. And you'll get some of those on your test, like Serratia has the red pigment, Micrococcus has the yellow pigment, Klebsiella has the capsules, Virilium has flagella, endospores of Bacillus and Clostridium. Some of the characteristics were obvious, some of them not so obvious. Neisseria, the Diplococcus with Fimbria. So some of those characteristics that we talked about in class will be on your test. And you didn't put those on your list. And your list is your study guide, so I would think you'd put those first, especially because in the directions, that's what I said. Make sure you put characteristics that were covered in class. If you don't believe me, go back and reread your instructions. Always about instructions, including those of you who are going to lab today. Read your instructions. Make sure you understand your instructions. Then proceed. Once you proceed, go back and check your instructions and make sure you're following all your instructions. Because you wrote up your whole bacteria list and then you didn't include the characteristics we covered in class. If you had gone back and reread your instructions, it would have reminded you to do so. So know you're going to have those kind of things on your test. Okay, let's talk about the degeneracy of the genetic code. First of all, our genetic code is called a triplet code. Why?
It has three letters?
Yes, because it has 3 letters. What are those letters?
It's not A, U.
What are the letters? A, U, C, G, T, okay, good. What are those?
Codon. Codon.
What are the letters?
Oh, you're asking for that.
What's this?
Nucleotide.
Good, a nucleotide. What are the letters?
Well, it's nitrogenous base.
Yes. The bases, nitrogenous bases are the letters. The letters are called purines and pyrimidines. The ones with a Y in them are pyrimidines. The purines those are DNA bases. And RNA, you also have uracil. Okay, your letters are nitrogenous bases. Every 3 letters is a what? And the codon is the genetic code for? Good. One amino acid. That's why it's called a genetic code. Every 3 letters is translated into an amino acid. So later we're going to come back and do the steps of protein synthesis. And we're going to do, just popped into my mind, just popped up. Oh, shopping list. This is why I write it on the board so I don't forget. Because sometimes it pops into my head, oh, we're going to do this, and then all of a sudden it's like left. I don't know where it goes, but sometimes I can catch it if I'm fast enough. Other times it doesn't occur to me until the next day or 2 days later. It's like, oh, now I remember what the hell I was talking about 3 days later. Okay, let's talk about the redundancy of the genetic code. The official name is the degeneracy of the genetic code. You might think of it as a redundancy. I don't know how to get rid of that thing. I don't know how to get rid of that side thing. Sorry. This is not the exact chart you have in your book in terms of the prettiness of it. But you do have a chart in your textbook. I think I mentioned it the other day, and Colton opened the page, and I don't remember what page it was.
202.
It's 20 what?
202 or 208.
202 or 208. Okay. This is the genetic code in mRNA codons. So every 3 letters in DNA is called a codon. Every 3 letters in RNA is still called a codon. Every 3 letters in tRNA is now called an anticodon. Remember anticodons from last week on your transfer RNA molecules? Sure you do. Okay, these are mRNA codons. So this is a working copy of the DNA. That's what mRNA was, the working copy of the DNA. The mRNA is made at the nucleoid. It's a working copy of the DNA. It's only single-stranded, so it only copies one side of the DNA. When it's done, it leaves the nucleoid, goes out to the cytoplasm, passes through a ribosome, where the transfer RNA molecules read mRNA codons, do complementary base pairing with their anticodon end, and bring the appropriate amino acid on their amino acid end to assemble a chain of amino acids. So, in mRNA codons, that's the photocopy of the recipe for a protein. So look at the mRNA codons. Up, let's say this second box, C and U. here. CUU equals leucine. CUC also equals leucine. CUA equals leucine, and CUG equals leucine. What the hell? As long as it's got CU, doesn't matter what the last letter is. That's the degeneracy or the redundancy of the genetic code. You can see it in this table of mRNA codons. Where I see it is in my turkey feet. So let's make some turkey feet. Alrighty? If the mRNA is CUU, then the tRNA would be GAA. If the mRNA is CUC, then it would be GAG. If the mRNA were CUA, then the tRNA anticodon complement would be GAU. And if it were CUG in mRNA, it would be GAC. And up here, it would carry leucine. You can see it on that table. There's redundancy there. I like to look at it in my tRNAs because it shows me this is going to complement the mRNA. So if this was your mRNA passing through a ribosome and these were coming in, this would be CUU, CUC, CUA, CU